Okay, so this is my Enzyme Lab Experiment. My name is Kamari Williams. This is for lab class. So introduction. Enzymes are biological molecules. They act as a catalyst to speed up chemical reactions within living organisms. Um, so again, they speed up their reactions through the catalyst. Basically, they lower the activation energy that is required for a reaction so that it can happen faster. Trypsin is our specific enzyme that we're using for the experiment. It is a digestive enzyme that breaks down proteins in our bodies. Um, so the reaction catalyzed by trypsin, it involves breaking down peptide bonds within proteins through process of hydrolysis. Trypsin can be a key enzyme in many organisms because it is able to break down proteins in our digestive system. It is found in the small intestines of many animals, including humans. It's often found in the humans and animals in a condition of pH between 7.5 and 8.5 and a temperature of 37 degrees Celsius. While in bacteria, they don't actually produce trypsin. It functions in the cell within, again, 7 to 8 pH and a temperature of 70 to 80 degrees Celsius. Our question for this experiment was, how does salt concentration affect enzyme activity of trypsin? Our hypothesis was that an increase in salt concentration will lead to a decrease in enzyme activity of trypsin because salt can denature the enzyme. Furthermore, our reason, um, an increase in salt concentration can affect enzyme activity due to the changes in enzyme structure because of the denature. Like proteins, enzymes structure determines its function, so a change in structure will ultimately change or defer its function. Um, our experimental design. So our experimental variable was salt concentration. Obviously, we used four different types of concentrations: 0, 0.0, 0, 0.15, 0, 0.75, and 0.6. Our control variables was temperature and pH. We used a temperature of 70 degrees Celsius and a pH of 8. We have 5 replicates for each of our concentrations, so we had 5 experimental no, we had 20 in total, and then our negative control we had 8, which was 2 per salt concentration. Again, our experimental design up there. For our procedure, we started off by adding each of the following into a centrifuge tube and mixing them well. So first we did 450 UL of reaction buffer, which was our salt concentration that we made. Then we added 500 UL of azo casein, which was the protein that we were using. Um, we mixed those together, and then finally we added our trypsin last. And for our controls, we added water instead of trypsin. Um, yeah, we added water instead of trypsin into the controls. Step 2, we incubated them for 10 minutes at our desired temperature, which is again 70 degrees. Once we finished incubating them, we put them in an ice bath for about 2 minutes. Once they were done with the ice bath, we added about 500 UL of TCA. Um, then we centrifuged all the tubes at 12,000 RPM speed for 2 minutes. Once basically centrifuging, it separated the supernatant and the pellet. We transferred the supernatant to a spectrophotometer cuvette, blanked the spectrophotometer with H2O, which is water, and then we read the absorbance of the supernatant at 440 nanometers and noted our data. So the why. Um, Starting off with why we chose this different salt concentrations. We chose them because in our last experiment, 0.3 proved our hypothesis the best, so we tried to find concentrations around that. We used 5 replicates because we wanted to produce more accurate results, and then we had a lot of replicates to be able to measure our variability later on when we were creating our graph and creating our standard deviation. Um, we chose our controls because we felt that trypsin worked the best at a pH value of 8 and 70 degrees Celsius, and we wanted to ensure that we were only seeing results for salt concentration and not for those two other variables. Our negative controls were used to determine if trypsin activity affects absorbance values. So we wanted to make sure trypsin was affecting it and not any other outside factors, which is why we had our controls. We used TCA to denature and precipitate undigested proteins. Um, so when we were centrifuging to precipitate the undigested proteins and have them as a pellet and have the digested proteins so that we can read them at the absorbance value. It also stopped our reaction to TCA so that it wouldn't keep on going, so that we could read it. Again, we centrifuged to separate the pellet, which was the undigested, and the supernated, which was the digested proteins. And then our substrate, we use azo casein, which is the protein, because that protein has a sulfanilamide group added to it, which creates the orange color, which we read it at the 440 nanometers. So, this is our raw data, as you can see. So we had the 5 trials, which is each of our replicates. We have all 4 of our different salt concentrations, and then we have our negative controls at the bottom. So each one, because we did have 2. So yeah, this is all of our raw data, all of the absorbance values that we collected. And then on this side, so we have our graph on the left side. So, we have our 4 different concentrations. The first bar, we have our experimental trial averages with the outliers. So in our raw data, we found that we did have outliers in some of the data points. So this is our averages with all the outliers, with all the data included. On our second bar, we have our negative control averages, so just the average of the negative controls. On our third bar, we had the experimental averages without outliers. So we took out the outliers in some of the data points. So these are the averages without the outliers that we calculated. And then beside them, we also have our standard deviation, which you can also see in the graph. So we calculated standard deviation for all of them. And then below that, we have our p-values. We have our p-values right there. Then on the right side, we have our graph with all of our standard deviations. So as you can see, based on the data, our highest concentration was 0.075, which is why when we were calculating p-value, we got rid of this and we calculated it based on the 0.075, and that's how we got our values. And as you can see, yeah, that's how we calculated them. And then our lowest value was actually 0.15. So highest was 0.075, our lowest was 0.15. So when it comes to results, the graph arranged based on the averages of the data without. So when it comes to results, the graph is arranged based on the averages of the data without the outlier included. This is just explaining standard deviation. So standard deviation is the amount of variability that the sample had between data points. So between the salt concentrations within them, how much variability they had. And so higher bars mean more variability, lower bars mean there wasn't as much variability. P-values, those were basically to determine whether or not our data was statistically significant. So we measure it based on the highest. So we compare the highest to the other data points, and then we get a number. And if that number is over 0.5, that means that they are not statistically significant. And then down farther, some sources of variability that we found in our data can be attributed to higher standard deviation. That can be attributed to higher standard deviation includes measurement error when pipetting, contamination from outside sources in between experiments, and complete randomness when it came to the results as well. Part 2. So this is discussing the actual data. So first, the experimental design and execution we felt was adequate to test if there was an increase in salt concentration and whether that would lead to a decrease in enzyme activity. Based on our experiment, our hypothesis was incorrect. The highest salt concentration we tested was zero point 0.6 moles, which was shown to have the second highest trypsin enzyme activity with a 0.2395 absorbance rate, and 0.075 moles had the highest enzyme activity with an absorbance of 0.3075. Meanwhile, our lowest enzyme activity of our experiment came from 0 moles at an absorbance of 0.1852, which was close to the lowest enzyme activity. Also, our t-test showed that none of our samples were statistically significant. That means that the data did not provide strong enough evidence to prove our hypothesis. Table 4 had an experiment. We compared um, our results to Table 4 because they also had salt concentration as theirs. So Table 4 hypothesis had an experiment where the range of lower to moderate salt concentration had the highest enzyme activation. Their results did not support our hypothesis. Moderate salt concentration enhanced enzyme activity while low concentration did not, as shown by the absorbance values and low p-values. At 0.3 moles, absorbance increased with 0.421 compared to 0 moles with 0.381, and their t-test indicated that the differences at 0.15 moles and 0.3 moles were statistically significant. Based on the results of Table 6, so we also compared our results to Table 6 because they did temperature and we were trying to see if our temperature choice was adequate enough. Our choices based on their results may not have been optimal for enzyme activity as they found higher enzyme activity at moderate temperature between 50 and 55 degrees, which means that a lower temperature may have actually improved our results and given us a higher enzyme activity. And then these are our sources cited.